angles. But today is the time where I'm most excited about because of the change that's happening because of the AI. Okay. I know that AstraZeneca is a household name since COVID, but I think it's good to give a little bit of an introduction of what company size is and what we work on. So we are a global science-led pharmaceutical company with a huge footprint. And I put a report from the 2024 annual report, which shows the revenue. It's about 54 billion. So this is not small. We are very, very large. And we spend about 13 billion on R&D alone, right? So R&D I will describe later in the session. And we have about 16,000 employees deployed worldwide, okay? One of the things that we are most excited about, and we speak about it in our annual reports as well, is something called as the bold ambition. So we have a bold ambition of 2030 that by then we will deliver 20 new medicine. This is a very ambitious target for any pharmaceutical to achieve. We want to be an $80 billion company, right? And most importantly, we want to be good to the planet, right? So these are all the things that we are working towards. And although I mentioned that we became quite famous since COVID, but our focus area is largely these therapeutic areas, right? So oncology is our biggest therapeutic area, followed by what is called as the biopharma. And these are things like cardiovascular disease, respiratory disease. But COVID, or when you call us the vaccine, right, well, actually falls into this area, right, which is just a very small fraction of what we do within the company. So if you are an uninitiated and you do not know what pharmaceutical looks like, right, maybe this slide will give you an overview. So pharmaceutical R&D is typically thought of as one thing. It's R&D. It's actually not that. It's actually composed of two distinct phases. One is called research, and the other is called the development, okay? Research is mostly about how you understand the disease, right? How do you find the targets? How do you test them on animals? But as soon as you move into the area of testing into the human, this is where the development kicks in, right? How do you manufacture things at scale? How do you launch the things out, right? That's all part of the development stage. Now, when R&D is thought about in this way, the easiest way to think about it is in terms of what disease we want to target. Let's say we're talking about pancreatic cancer, right? That's how typically a research question is started. You look at what the underlying mechanism for the disease looks like. This could be a target, right? Then the scientists go and they develop medicines for that, right? Or molecules, they are called, right? Think of them like a lock or key, right? That you're trying to unlock. Then you test them on the animals, right? To check that they are okay to use. Then you manufacture them at the scale. But when you go into the development phase, this is where the drugs are tested in human trials. This is where the biggest regulatory burden comes on us, right? And once the drug is successful, it's safe and it's efficacious to use and effective to use, it gets sent to the health authority for approval, and this is where you get the medicine. Now, this process here is long. It's hard, and it takes huge amount of effort to do that. And just to give you an idea, I pulled some stats from AstraZeneca's data, right? And it shows here that from the left side, all the way to the right side where you have that medicine launch, it takes on the average 15 years to do it. 15 years to launch a drug from the research all the way to the down on the average, right? It takes about 3 to 6 years time in the research, means about 40% of the time that we're spending, money that we're spending in the research, and about 7 to 9 years in the development phase, okay? And on the average, it takes about $2.5 billion to launch a successful medicine at the end, right? So we are working on these innovative therapies here, right? We don't have to worry about what those are, but the idea here is to show that when we start this journey, there are huge amount of risks involved. The risks are all the way to the end, and this curve here is actually a cone of uncertainty, right? The earlier you are in the phase, the most uncertain you are. And as you go down the lane, as you start getting better data around your molecule, the certainty keeps on decreasing, and you can finally launch a molecule or a drug, right? But that's not the end of the story. You can also have a situation where you launch something, and you start realizing that there are bad adverse events. There are medicines which have been pulled out from the market, although they've been approved. just because there were bad side effects of that. So there is always a risk attached to this process. Essentially, what I'm trying to say is that this is a high-stake industry that we are working in, and a lot of investment is needed here. Okay, now this is the most I'm going to talk science today. Okay, so even if you don't have a science background, I think you will certainly appreciate this. There is a concept in science that is called as biomarker. Okay, that is integral and a very important concept when you look across the research and development, and this is used for a lot of scientific innovation work. When you think about measuring blood pressure, right? That's a biomarker for your cardiovascular health, right? If you measure the blood sugar level, well, that's a biomarker of your diabetes level, right? Anybody is a fan of Marvel movies, right? When you get a mutation in the gene, you become a superpower. You get a superpowers, right? You become a superhero. But in reality, when you get a mutation in the gene, it can be a potential disease-causing problem, right? That mutation in the gene is actually a biomarker, okay? So this notion of biomarker is used across the whole of research and development. It is used for testing whether a person has a certain disease, or they will respond to a therapy that I'm building, or what their prognosis is gonna look like. And this is essentially what makes the scientific innovation possible, okay? Now the problem with biomarkers is that I can go from things like wearable devices, I can go from things like blood pressure measurement, or I can go to the microscopic level, and they are notorious for generating huge amount of data, right? You see where I'm going with this? Well, this is where your AI comes in, right? Where you can apply these computational methods on this to find novel stuff. Let me talk about something else, which is called as FAIR. Anybody familiar with this term, FAIR? All right, few. Okay, so this concept came out about a decade ago in a paper published by these very brilliant authors. And essentially the idea of FAIR is that how can you make the scientific data, the stewardship of it, the management of it, really manageable to some well-defined principles, right? And the FAIR stands for something like, okay, the F here is findable. I can find my data. If I should be able to find the data, it's good. It means that I'm not storing the data on my laptop. I'm not storing it in some corner of SharePoint site, okay? Accessible means that I can access the data with the right governance around it. Interoperability means that I can pull the data from multiple sites and combine them together. And then R, which is the reuse, meaning that I can always reuse the data without making my own copy of it. Right? Does it sound familiar? People always tend to make copy of the data, right? Well, FAIR discourages all of that. So this paper also says that these are the good principles. If you can carry them out, I think you have quite a mature data stewardship, right? But there is also two aspects that I want to call out. One is that, although people talk about this FAIR concept for data, it's equally applicable to the analytics, okay? I could be building notebooks, I could be doing informatics analysis, but I could apply the same thing there. The second thing this paper calls out is that this FAIR principle enable access to the data, not to the human, but also to machines, right? So if you have things like agents, right, they can actually leverage some of these things to do some cool stuff, all right? So over many, many years now, I think we have spent close to a decade on this. We have developed a very mature data strategy at AstraZeneca, and this is what roughly the architecture of that looks like. So we have a lot of data sources, right? They're coming from the labs, they're coming from the instruments, they're coming from our clinical trials. They go into what we call as our system of records layer. These are your operational stores, right? These are your transactional systems, right? Where the data gets stored. But then we apply a data mesh architecture. The data mesh say that, well, you can federate the building of the high quality data out to the domains, right? And we have many, many domains. So I put some exemplary domains there. And these domains are essentially taking the data from those system of records, which are represented in ping, doing the analysis and putting them in a very analysis-ready, high-quality data products at the top. Okay? Does that make sense? Right? And every domain owns that. We federate the governance of that data to as much as possible because the ownership of that domain determines the best way to federate it with some centralized controls. And then we have some linked data, which is the graphs built on top of that, the knowledge graphs, right? And once you have this high quality data that has been built in this way, you can start building different types of applications on top of it, or the AI-based applications on top of that. Okay, so we have spent a lot of years building this. We have built a data-first culture. It took us a very, very long time. But I want to show here later on that just because we have made this significant investment in this over a number of years, right, is paying the rewards or dividends a lot for us now, especially when we're moving into the agentic space. This slide, this is one of my favorite slides, right? It's a very simple slide, but it shows the stuff that we heard about in the beginning, right, in the keynotes and the other session, that the FAIR concept applies on the data, but it also applies to the analytics, right? I should be able to find the analytics that are conducted by my colleagues, right? Of course, with the right governance around it. I should be able to access it if I have the right access controls around that, right? And we are investing very heavily with Domino here, right, to enable this vision of FAIR. And if you have the FAIR data, and if you can verify your analytics, this leads to what we call as the trusted science, right? I can trust the science of my colleagues, right, which I can even use for submission out to the health authorities, okay? So this is an extremely, extremely important concept for us. Okay, I'm gonna switch gears now and gonna talk about foundation models, right? Exciting stuff, right? Data is boring now, right? If you look at how the field of AI has evolved, we had traditional machine learning, right? Every domain, every pipeline that I want to build is going to be very task specific. It used to be like this. I will build a pipeline that will do just the text summarization, or something that will do the detection of nucleus in the cell images, right? There used to be a team that will build those models out, right, and manage them. You will have the MLOps for every left to right models that are going to be built. And all these machine learning models are doing is that they are simply doing pattern recognition. But in the last few years, foundation models emerged, and they drastically changed the landscape. And what these foundation models are, that they are taught on or trained on this massive internet level data sets, and out comes these huge models, right, that can do something magical, okay? And their magical properties is what is called as prompting, right? This is what gives you the economies of scale. So I can just use the same model, and you can use it for your intent, I can use it for my intent, same model, but different intents, yeah? This is the economy of scale. This is what enables my 10-year-old to write code, right? And my 80-year-old dad can use the same thing for doing something cool, right? And this is what democratizes access to the foundation model, to the AI. The other characteristic of these foundation models is that they can be fine-tuned, which means that I can take them and fine-tune them for certain downstream tasks and make them more specialized if I want to, based on my internal data sets, right? So the first one where you're just prompting and stuff, this is something called the zero-shot learning. The other one is called the future learning, when I'm doing the in-context for these foundation models. And one of the cool things is that while in the traditional machine learning model, they were just doing the pattern recognition, in the case of foundation models, it can actually do the reasoning, right? If you use like something like DeepSeek, right? Or if you look at some of the recent innovation in, let's say, ChatGPT, right? You can see it reason, right? People can question that, but it's reasoning in a way, right? And that's an important characteristic. It gives you the traceability of how it's thinking about that, okay? Let's take it to the next level. Let's talk about LLMs, right? Large language models, where they are nothing more than just a type of foundation models, right? Simple as that. So it's generative AI sits somewhere at the bottom. But I can take the concept of LLM, right, and augment it with some things extra and make it into an AI agent. It's a very, very simple way of thinking about the AI agents. I can take the large language model and attach it to a tool. If it can talk to a tool, which could be an API, it could be a software. If it has certain memories, it can remember the context that I'm talking about. If it has the ability to reason, this is what gives you an AI agent, okay? So now you're graduating from an LLM to doing something which is a little bit more sophisticated. But if this is sophisticated, let's make it a little bit more fun. I can build all of these agents and attach them together, right, in some sort of a topology. And this is what is called as the agentic workflows. Does that make sense? So I can talk to these guys, right? These agents can talk to each other, for example. I can talk to those agents, and through some sort of a clever orchestration, they can accomplish some very, very complex tasks. So as you're moving from left to right, you have a higher level of autonomy that you can achieve. And I will try to show you later on that how we are applying this pattern in our R&D research. This is how we have been thinking about the AI agents now, right, in terms of their autonomy. So you have level 1 to level 5. Level 1 are simple things like prompt-driven tools. If you're familiar with ChatGPT 3.5, you can just talk to it. It has no context of memory. It has no reasoning ability. Well, really, something as simple as that is level 1. Level 5 is fully autonomous. This is something hypothetical right now. If you think about Terminator, Skynet, well, that's level 5, yeah? Now, we are somewhere in between that, okay? So if you think about tools like GitHub Copilot Studio or LangChain, right? That's somewhere in the level 3. And you can see that as these levels are increasing, the tools that I mentioned or things that I mentioned in the previous slide, they start to appear or they become more mature. Okay, so we are not there at level 5. I think we are somewhere there between level 3 and level 4. Within AstraZeneca, we are operating somewhere around level 3, I would say, right? Or getting to level 3. Okay. Okay, so I'm going to bring us back to the biomedical side or the science side of things. And I hope you will appreciate this one here. So this is a pull from a paper. And I love this paper because it talks about the evolution of AI or the models, right? Mostly using the digital images, right, as an example. But I think the concept is very well suited to explain where the things are going here. So in early 2000s, people will build machine learning models. So scientists will build these machine learning models with handcrafted features. So somebody will go and handcraft the features, right? Then feed it into the model, and the model will be trained on that, right? This was like early 2000s. Then in 2012, you had this deep learning model that started coming in. And what these deep learning models did was that, well, you don't have to train anymore, you don't have to do the feature extraction. You can work with very large label data sets, and the model can learn the hierarchical features within the data sets, right? And this is all very good. This is in the context of like a supervised machine learning, all good. But then in early 2000s, people started playing with the notion of multimodal data set, right? So machine learning is cool. What is cooler is the multimodal data set, models. And what multimodal data sets models do is that they can take the images, they can take other type of data sets, right? And try to merge them together to make sense of what the response should be, okay? So here we use an example of genomics data set, imaging data sets, patient data sets, right? And I can see that multimodal data set models are built that can predict the response versus not response to a certain therapy, right? So you can see that the models are becoming a little bit more generalized as you're moving from left to right. Then somewhere in 2023, the foundation models started appearing. And these are these internet scale, massive foundation models that are trained on diverse, heterogeneous data sets. And again, it's all moving towards the generalizable models. And the aim there, according to this paper, is to build this one meta generalist model that can be very good in doing various downstream tasks. So it can predict, for example, the drug response. It can predict various types of cancers. It can predict the radiology assessment, right? So this is the end dream. I think we are somewhere there in the foundation model category. Okay, so if I pull all of these views together, right? What we have found is that when we want to do a scientific discovery in pharmaceutical R&D, it always requires this multimodal data set. Multimodal data set means that I'm looking at the images, like pathology images. I could be looking at radiology images, X-rays and MRI and such. I could be looking at molecular data such as genomics, right? Or clinical data sets, or I'm looking at the external data set published by the universities. But in order to work with this data set, I need a multidisciplinary team. I need a statistician, I need informatics, I need data scientists, I need lab data scientists, right? In order to make sense of things, right? But in addition to those two, we also need something called as the toolbox. So these are your traditional software or tools that we use across the spectrum. You have genomics-related tools, you have pathology-related tools, things that we have built over many, many years of research or time, and we use them quite heavily. So the combination of three is what enables this scientific discovery. So we went around asking these questions of the scientists. We did a big survey last year. We asked, I think, close to 100 scientists within AstraZeneca on how the biomarker research is going to look like in their area, what type of questions that they are looking for, etc., and what sort of impact if we can answer those questions very rapidly. And what we have found is that there are, in terms of complexity, we found out that there are questions which are very, very simple, right? And the questions which are very, very hard. The hardness of the question depends on how big the data set is, how complex the problem is, how many of the multidisciplinary teams that you will need to bring together, and what the impact of those questions is going to look like. So the green represents something simple, red represents something hard, and something which is very trivial could be as simple as just looking up a record in a database. Right? Something complex could be a multi-step, multidisciplinary team coming in to solve the problem over a number of years, right? And it's a very good characterization of how the biomarker research, if we were to make it mature and make it a little bit more systemic, this is what it's gonna look like. So this is what we have tried to solve using this agentic thinking that I have been talking about here. So a scientist can ask a question. How do patients with KRAS mutation respond when treated with durva in solid tumors? You don't have to understand the science behind it, but I think you can understand it's kind of like a simple to medium-sized question, complex question that a scientist would typically ask. But when we apply this in the context of agentic thinking, right, this is what happens. So I will build an agent that will first do the question decomposition. What it will do is that it will look into the question and try to unpack it. It will say that durva is actually a name of a molecule. It's a cute name for one of the molecules because we are terrible in naming the drugs, right? So durva is a cute name of how we name the molecule internally, right? So it will try to disambiguate that term. It will ask the question, oh, you're looking for a KRAS mutation, but where is that mutation data coming from? What does the term respond means, right? You and I know what that means, but the agents do not know that, right? What does the solid tumors mean? Where is the data coming from for that, right? So this is the type of things or unpacking of that question or disambiguation that this agent is going to do. Then we have a planning agent that takes that problem, right, and identify what are the right agents that I have access to that I can bring to bear to answer those questions. Okay, and there you can use some of the some patterns like React and such. But what we have built are these what I call as the knowledge agents. Okay, these are the list of knowledge agents that comes into play, that the planning agents bring them alive from the, let's call it, the marketplace. Okay, so you can have like a biomarker agent, right? And what that biomarker agent is doing is using one of the big foundation models, right, that are available. Or it could be using something else that we have built internally and querying the FAIR data. Remember the FAIR data we talked about? Well, this is what it's doing, right? We also have, let's say, a literature agent that goes out and finds information that I need from the publicly available information. I can have a drug agent, right, that understands the chemistry of the molecule, right? So planning agent essentially is pulling those agents out, orchestrating the questions out to them, and getting the answers out. And once you get the answers out, they actually go to some resolvers towards the end. So you have like evidence synthesis agent, which takes all the aggregate of all of these knowledge agents and synthesize the information. It can do some sort of a debate in this area and try to come to a consensus. And then you have an answer generation agent that, based on my knowledge, based on my context, right, gives me the appropriate response. So, for example, if a scientist in the lab is asking a question, there might be certain things which are more relevant to them than the other. So the answer agent has that kind of an awareness of the user persona. So before I move on, I cannot emphasize the importance of this FAIR data, okay? Because if you get this right, if you have the right governance around it, everything starts falling in place very easily, okay? Well, I say easily, but it becomes relatively easier. Okay, if you don't have this instead. All right, so if I pull all of those views together, this is what our view is. So we are working with a huge amount of a data set. A lot of this is proprietary. The data that we generate from our preclinical research, from our clinical trials, we source in a lot of public data sets because this is like a treasure trove for us, right? We generate a lot of data from our collaborations, right? We also generate a lot of synthetic patient data sets, especially when it comes to anonymization and such, and we don't want to use certain data sets for various, let's say, regulatory reasons, right? But now our focus has largely been in bringing in various types of foundation models. So there are some that we are building internally. There are some that we are developing as part of the bigger collaboration. There are numerous foundation models which are in the open source community right now, right? And then stitching them together through some sort of an agentic framework. So you can have like a RAG-based, your traditional RAG-based approach, right? Or you can do the React-based things, etc. We're not doing so much in the fine-tuning space right now because it creates certain complications. But the idea here is that with zero shot and few shot, we can get quite far from the research perspective. The other thing that I want to mention here is the use. I use a term there on the top that is called a single pane of glass. So all of these things are cool, great, right? But if you don't have a very good user experience, right, this is going to fall really bad, right? So single pane of glass is our experience layer that allows users to discover models. It can discover the agents, right? And I can compose something very, very interesting using that. So our fair data is kind of being data sources are being surfaced through the FAIR data set, and Domino is providing currently our environment where the models land, and we can try to build the RAG architecture on top of that. Okay. All right. So let me conclude here. All right. So the 3 points that I'm going to take, I want you guys to take away. One is that FAIR has been really instrumental for us, right, as we are trying to build these agentic AI, right? So we started building a lot of our assistants, let's call them this way, in early 2023, just because we had a very strong data foundation, right? So we quickly built something called as Research Assistant that can query literature data set. It can query a lot of CRISPR and a lot of lab-based data sets easily. Then we built something called as a Development Assistant, right? Last year we launched it, we productionized it last year, and this allows clinicians, for example, right, or people working in the ClinOps space to run queries on the clinical data set. So I can use it to design clinical trials. I can use it to optimize the ClinOps process. I can run queries on the SOPs, clinical SOPs, right? The boring stuff, right, that nobody wants to work on. But recently, I don't know whether you heard the news, AstraZeneca entered into a huge partnership with Tempus to build this big biology foundation model, right? That we are very excited to bring it in a couple of months and make it like one of our universal model for oncology. And the idea here is that at least with these sort of technologies in place, we can ask at least trivial to medium type of biomarker questions. The second point I want you to take away is that this is nothing, there are so many risks attached to this one, right? So if you plot these risks versus your appetite to do innovation versus automating the routine tasks, you'll find that where you have the low risk innovate or low risk to routine tasks, these are your agentic AI opportunities, right? You can do a little bit in the routine tasks and high risk area too, but you need to be careful with that. The third thing here is that while you can have fair data, you can have all sort of regulations in there, human in the loop is indispensable. Okay, you cannot eliminate human currently. And this is what, and the idea here is that just because the way that the LLMs operate, right, the way that they can hallucinate, I think they cause some of the challenges for us. So human in the loop is essential for us. All right. So this is my last slide. Happy to take any questions.